Hello everyone, and welcome to the screencast for Chapter 5, the cell cycle, mitosis, and meiosis. As we learned earlier, human cells and all cells are very, very small. But think of it, you started out as one cell, and now you're composed of hundreds of trillions of cells. These cells are very small, but they must be dividing all the time. And that's what Chapter 5 is about, cell division and the cell cycle. In the first section of this Chapter 5, we're going to look at the difference between cell division in prokaryotic, simple, small cells like bacteria, and eukaryotic cells. Your cells are eukaryotes and they have nuclei in them. That's the big difference. One is much smaller, the prokaryote, the bacteria, much smaller than eukaryote cells, which are much bigger. We're going to look at the phases of eukaryotic cells, and this is the specialized vocabulary that we'll be learning. Cell division is a process in which one cell, called the parent cell, divides into two new cells, which are referred to as the daughter cells. Cell division is simpler in prokaryotes. Remember, anytime you see the word prokaryotes, kind of think of bacteria when you see that word, than eukaryotes. And that's what we'll take a look at next. When prokaryote cells divide, it's referred to as binary fission. So if you take a look at these two diagrams, this is binary fission in a bacteria cell. And read through that, and you'll be able to understand that it's a much simpler process than what you're going to see in a few more slides. Here we see the steps of binary fission, where we have the mother cell, and in it, it says the cell. In the second diagram right here, it says the cell elongates and replicates. So remember, what replicate means is that it's copied. All the initial DNA is copied before the cell begins to split into two daughter cells. However, the cell cycle in eukaryotic cells, the cells that have a nucleus, is a little more complicated. And this is a stylized version, a diagram, which represents the cell cycle in eukaryotes. And imagine the two daughter cells form right here. We have the resting phase, the first gap, the synthesis phase, the second gap. But the part we're going to emphasize right here in mitosis is when the cell actually divides in half. And that's composed of prophase, metaphase, anaphase, telophase, and cytokinesis. These are the steps as the cell divides. Remember, these are the eukaryotic cells. They're bigger, they have a nucleus, than the prokaryotic or bacteria cell. This diagram from your textbook talks about the cell growth checkpoints. Number 1 occurs toward the end of the gap phase. Part 2, DNA synthesis. This is when DNA starts to grow. And finally, point 3, the mitosis checkpoint. Make sure you're familiar with these three major checkpoints, and make sure you're familiar with this diagram, especially the mitotic phase. One of the things we want to mention right now is cancer. And what happens with cancer cells is they lose their function and they basically turn into cancer cells and grow uncontrollably. They speed up their cell cycle, they grow much faster, they still need all the nutrients and everything, but they don't perform their own function, and they grow usually, they form a tumor. A tumor can be small, it can be large, it can be in the bloodstream, it could be in a different type of organ. So cancer is uncontrolled cell growth. In section 5.2, chromosomes and mitosis, what we're looking at here is the role of chromosomes as they double their DNA and break into the steps. Notice the objectives for this part are describe chromosomes and their role in mitosis and outline the phases of mitosis. So if you look at the vocabulary, you're going to learn new terms such as anaphase, centromere, chromatids, chromatin, chromosomes, gene, homologous chromosomes, metaphase, prophase, and telophase. So take a look at this. This is an important diagram that might be on your quiz. This is mitosis in eukaryotic cells. And what you want to look at right here is you want to look right here at the nucleus. So this is the nucleus. This is where our DNA is. These are all the other organelles in a stylized cell. Watch here and right here from this phase into prophase. What we have happening is the DNA, the chromosomes, are doubling because we want a complete set of chromosomes in each of the new daughter cells when we end up splitting over here. But first we have prophase, and that's when the DNA doubles itself. Then the DNA in metaphase all lines up right in the center part of the cell. And slowly, those chromosomes in anaphase are pulled apart and then repositioned in a new nucleus in telophase, and then in cytokinesis, the cells actually begin to divide into the two daughter cells. And the whole cycle will start again. That's why it's called a cycle. It just keeps going and going. Remember, mitosis is cell division. So I wanted you to see a microscope slide of this actually happening in cells. This is a snapshot. This is a frozen piece of onion root tip. It's been dyed blue so that you can see the chromosomes much easier. So I'm going to look at a couple different cells. Now, many of these cells are in interphase, like this cell right here. But look at this cell right here. This cell right here, all the chromosomes are lined up. That's metaphase. Oh, look at this cell over here. The chromosomes are being pulled apart. That's anaphase. This part is just before metaphase when the chromosomes have doubled. That's prophase. So this is a snapshot. Lots of different cells here. We have over 100 different cells here at different parts of the cell cycle, different parts of the mitotic cell division. Here's some terms from your textbook that you should be familiar with. Centrioles are bodies that attach using these microtubules onto the chromosomes. Here are chromosomes here, and with microtubules connected to the centromeres, that's what's going to pull these chromosomes apart. Half of them will go into the part over here, which will become a daughter cell, and the other half will become into this area and become a daughter cell. In section 5.3, which is called Reproduction and Meiosis, you'll notice the term meiosis. So meiosis is different than mitosis. Most of the cells that you undergo cell division in is mitosis. However, in human beings, there are specialized sex cells that undergo meiosis, because at the end of the meiosis process, we don't want a pair of chromosomes in those cells. We only want one single chromosome. We'll talk about the reason for that later. So in this lesson, we'd like to compare and contrast asexual reproduction with sexual reproduction, give you an overview of sexual reproduction and the phases of meiosis, and explain why sexual reproduction leads to variation. That's a very important term in offspring. The vocabulary here is asexual reproduction, crossing over, diploid, egg, fertilization, gamete, gametogenesis, haploid independent assortment, life cycle, meiosis, sexual reproduction, sperm, and zygote. Make sure you read and understand the difference between asexual reproduction, which involves a single parent and results in offspring that are genetically identical to each other and to the parent. So there's no mixing of genetic material in asexual reproduction. Sexual reproduction, on the other hand, involves two parents. They refer to the diagram here. In sexual reproduction, parents produce reproductive cells called gametes. So in human beings, if you're a guy, you're producing gametes, which are called sperm, and if you're a girl, you're producing which are called eggs. And both of these gametes have what are called haploid cells, and that means they have only half the number of chromosomes. So this is why we want our reproductive cells to have half the number of chromosomes, because when they come together to form that first cell which is referred to as a zygote, they will have pairs of chromosomes. They will have, in the case of a human being, 23 pairs of chromosomes. We talked about binary fission before as far as bacteria and prokaryotic cells. Here in your book in 5.13, we talk about binary fission with single-celled organisms, that they're the ones on the left, like the amoeba, like the flagellate here, and the paramecium. But we also have multiple celled organisms like these, also producing asexually. This diagram gives you a quick overview of meiosis. Notice what happens here. When we come down, as we go through the cell division, the cell then divides again right here in meiosis 2. So meiosis 1 is sort of like mitosis, but then meiosis 2, it puts not paired chromosome in each daughter cell, but single chromosomes into each daughter cell. Notice that up here we had paired green chromosomes, but down here in the daughter we only have one. This might be a sperm if you're a guy, if you're male, a human being. It might be an egg if you're female. Also notice that sometimes there's a mixing of genetic material on that single chromosome. One of the biggest examples of the advantages in sexual reproduction is the amount of variation that you get. So when you read this section, make sure you know what crossing over is, make sure you know what independent assortment is. But look at this last line in your textbook. All of these mechanisms, and what they're talking about there is sexual reproduction, genetic variation, working together result in an amazing amount of potential variation. Each human couple, for example, has the potential to produce more than 64 trillion genetically unique children. No wonder we are all so different. But keep in mind, right now on the planet, we have over 7 billion people, and yet we have the potential to produce over 64 trillion genetically unique children from just one human couple. It's really interesting. So make sure you read all of Chapter 5, go over all of the vocabulary, and good luck on the quiz. If you have any questions, we'll be able to talk of them at our review sessions.